my name is Helen, and actually, I think there is a typo on the program. And unfortunately, I'm only going to be able to talk about my project on the characterization of ATP release by Salmonella and E. coli, so no biofilm, unfortunately. But, so yeah. So here's a quick overview. So everybody has heard of Salmonella. It's gram-negative. It's in reptiles such as snakes and turtles, and people have these reptiles as pets, and sometimes they are carriers of Salmonella. So if they play with them, they can get Salmonella poisoning from their pets. Also, it's really common that when they eat food that hasn't been stored properly or cooked thoroughly enough, such as eggs or poultry, that can be carriers. They can get Salmonella from that too. And Salmonella, as I mentioned, causes food poisoning, gastrointestinal inflammation, bacteremia, fever, and sometimes even death. There's about 1.4 million illnesses and more than 400 deaths in the U.S. alone each year. So gram-negative bacteria have inner and outer cell wall, separated by periplasmic space and a thin layer of peptidoglycan. And in their cell wall, they have different proteins, such as transmembrane proteins, which include porins, and that's shown here in orange. And then these are other proteins. So my project focuses mostly on porins. And porins are actually proteins whose molecules can form channels and allow hydrophilic molecules to pass through the membrane, which otherwise is hydrophobic. So an important molecule is adenosine triphosphate, which is hydrophilic, and that's also known as ATP. So adenosine triphosphate, also known as ATP, is the energy currency of the cell in all organisms. We have previously detected ATP in the culture medium as Salmonella growth. So for example, if this is a single cell of Salmonella and it grows, we see a lot of ATP that are not in the cell and actually in the medium, and that's shown here with the yellow circles. And we don't really, there's some unanswered questions. We don't really know why they're releasing this ATP. It doesn't make a lot of sense because if ATP is energy, it would be more beneficial for them to keep it than to release it. One of the things that might be a possibility was that they would use it to make biofilm, and that would allow them to survive more adverse conditions, like with antibiotics present or like low pH. But we're still not really sure. And we're also, another question that's unanswered is how it's being done. We don't know if it's through like certain proteins in the membrane, but we do know that deleting certain genes that encode for porins have shown that there is a decrease in ATP release. So this leads us to suspect that maybe a porin may be in control of releasing ATP. So my project focuses mostly on porins and their potential role in ATP release. So the way I approached this was by performing a genetic study for porins and ATP release. We obtained different porin mutants from E. coli that had a deletion in different genes that coded for different porins. Then we measured the ATP release for each of these mutants, and we also measured the ATP release for the wild type without any deletion mutants at all. And we compared the two so we can see which mutants were defective in comparison to the wild type. So here's the ATP release assay. So here's how we did it. We grew each mutant overnight in Liria bertani broth, which is just a nutrient-rich medium that allows bacteria to grow more efficiently. And then at 3, 6, and 9 hours, I performed an assay called Bactiter Glow Microbial Cell Viability Assay. And basically what that is at the time points that I mentioned, 3, 6, and 9 hours, I would take the bacteria and spin it down and get the super mutant. Then I would place the bacteria into the wells of a plate and add a reagent called Bactiter. And basically what Bactiter does is it kind of interacts with ATP, and when it does, it luminesces, and that can be picked up with a spectrophotometer. And in addition to the mutants, I also added different concentrations, or known concentrations of ATP into the wells, and I also added Bactiter to that too. And this is because since I know the concentration of the ATP that I put in, when I get the luminescence values, I will have a luminescence value to compare with the actual concentration I know. So So I can make a standard curve and use the luminescence values from our mutants to get the concentration of ATP in the supernatant. So here is the ATP Release Assay data. If you look at this is wild type, this purple one right here. And most of them generally have the same shape, like 3 hours, not very much, and then at 6 hours, it's kind of spike, and then it goes down around 9. But if you look at the blue line, that's for a gene called OMP-F. And in that, if you look at that, it doesn't look the same as the other ones. And at 6 hours, it's pretty low compared to the other ones. So that led me to suspect that maybe OMP-F, the OMP-F gene, plays a role in ATP release. So now that I have a mutant that I suspect may be controlling ATP release, I want to complement that mutant with the wild type gene, because if I complement the mutant with the wild type gene and the phenotype is restored, then that implies that the gene plays a role in ATP release. So the way I did that was first I purified a plasmid, and that plasmid is called PRB3273C. And a plasmid is basically just a circular piece of DNA. And I also did a polymerase chain reaction to make many, many copies of the wild type gene. And then after that, I digested both the plasmid and the wild type gene with a restriction enzyme called HindD3. The reason I digested them with the same enzyme is so that they would have compatible ends and I can ligate them. So first, to make it more simple, I actually labeled each side A and B. So when we ligate the gene and put it into the plasmid, it can ligate in either direction. It can be like A, B, A on the left, B on the right, or it can be B on the left and A on the right. And there's no selective advantage to having either orientation. So we don't know which direction it went in. We'll solve that. I'll explain how we solve that later. But so we expect that there should be 50% of each, since there's no benefit to having one over the other. And so after I ligated them, I transformed the ligated DNA into E. coli competent cells, and then I plated those cells on agar. So after the cells grew, I wanted to screen for cells that were screen for recombinants, so cells that picked up the plasmid that had the wild type gene in it. So I did this by doing another polymerase chain reaction using puck forward and reverse primers. And basically what those do is they will cut at the red line and amplify the gene that's in between them. And so if the plasmid did take up the wild type gene and it was successfully picked up by the cell, then the gene in between will be amplified, and I can visualize that on an agarose gel. But if there wasn't a gene that got picked up, then there would be nothing to be amplified in between the two sites, so it would be blank. And so here on the right side is an agarose gel, and in lane 1 is a ladder, and that's basically just a DNA of known lengths that we load onto the gel, so that when we visualize the gel, we can see all the different lengths and compare our mutants to the length of the ladder, so we know how big our mutant DNA is. And so if you look at columns 2 and 5, there are bands there. There's 2 bands, and the other ones don't have any bands. So this implies that column 2 and column 5 have successfully been ligated and they have the DNA. But at the same time, the other lengths don't have any, so those colonies haven't picked up the DNA. And so now, even though we were able to successfully have the wild type gene go into the plasmid, we still don't know whether the gene is in the AB direction or the BA direction, because even though our primers can cut on either side, they don't distinguish between the correct orientation. So the gel will show us that we have this DNA here, but it won't show us if that's exactly what we want. So to solve this, what we did next was DNA sequencing. So we sequenced the DNA using a Porins reverse primers to check the orientation. And basically, that's currently what I'm doing now. And basically what that is it allows me to see the sequence of the gene, and that is just the pattern of adenosine, G, and C nucleotide, and whether it's in the correct orientation or not. So I have the actual DNA sequence of the wild type in the correct order. So once we obtain the sequencing from the facility, we can compare the sequence from that to the actual correct wild type one that we want, and we would know if it was the correct orientation or not. So future directions. For right now, I'm still working on transforming the plasmid containing the wild type ONBEF gene to complement the ONBEF mutant in E. coli. So once I can confirm that the gene is in the correct orientation, we can complement the wild type with the mutant and see if it would restore the phenotype. If it does, then, like I mentioned, it would imply that ONBEF is importing an ATP release. Then I will complement the ONBEF mutant with mutant alleles containing substitutions in charged amino acids. We're going to change aspartic acid to asparagine, glutamic acid to glutamine, or one of them, not all of them, but yeah, and 3 arginines to alanines. And changing these amino acids at important positions will change the size and the affinity of the channel, and I can study how this would affect ATP release in this gene. And of course, whenever I want to study the ATP release, I would always use what I mentioned earlier, the whole back titer ATP assay to quantize the amount of ATP that is present. And yeah, so I want to take this time to acknowledge the staff, Professor Liu, for being a really good mentor and teaching me so much and being really patient. Max, Chow, and I want to thank the Surf Rose Foundation for all the funding, and the staff, Nathan and Leah, for their guidance.